Hello and welcome back to NEET Prep Go. So last time we did animal husbandry part of strategies for enhancement in food production. Let's revise fast and then we'll move to the plant breeding part. So with ever increasing population, enhancement of food production is a major necessity. Yes. And biological principles as applied to animal husbandry and plant breeding have major roles in our effort to increase food production. Several new techniques like embryo transfer technology and tissue culture techniques are going to play a pivotal role. So we are going to study about this ahead. So firstly, animal husbandry. What is animal husbandry? That is the practice of breeding and raising livestock. Agricultural practice of breeding and raising livestock is called as animal husbandry. It is a vital skill for farmers and it is as much as a science as it is an art. This we had learned last time. Now, what all animal husbandry includes? It includes with the care as well as breeding of livestock like buffaloes, goats, cow, pig, horse, cattle, sheep, etc. And these all are useful too. So, animal husbandry deals with the care and breeding of livestock like buffaloes, cows, goats, sheep, pig, camels, cattle, etc., and horses, etc., that are useful to humans. It also includes poultry, fisheries, and poultry farming and fisheries. Fisheries includes the rearing, catching, and selling of fishes, as well as mollusks, that is shellfish, and crustaceans such as prawn, crab, etc. The seafood that all you eat. Now, since time immemorial, animals like bees, silkworm, prawn, crab, fishes, birds, pigs, cattle, sheep, camel, etc. have been used by the humans for products such as milk, egg, meat, wool, silk, honey, etc. It is estimated that 70% of the world's livestock population is in India and China. Livestock population is in India and China. However, only 25% of the world farm produce that is contributed. So the productivity per unit is very low. This is an important line. Yeah. So it is surprising to note that the contribution of the world farm produce is only 25%, therefore the productivity per unit is very low. And in addition to conventional practices of animal breeding and care, newer technology has also been applied to achieve improvement and in quality and productivity. Now, a professional approach to what have been traditional practices of farm management gives the much needed boost to our uh, food production. Okay, so firstly, dairy farm management. Dairy farm management, dairying is the management of animals for milk and its products. This we had for human consumption. This we had read last time. And in dairy farm management, that is, we deal with processes and systems which increase the yield and improve the quality of milk. So milk yield is primarily dependent on the quality of breeds. This is very important line. Milk yield is primarily dependent. So of all the so question will come that uh, milk yield is dependent Primarily on what? There will be hygiene, there will be in the options there will be hygiene, the disease resistance, and yeah, good maintenance. But you have to primarily, if they say that is milk yield, overall it depends on all those factors, but primarily it is dependent on the quality of breeds in the farm. Selection of good breeds have high yielding potential under the climatic conditions of the area, combined with the resistance to in diseases. So for yield potential to be realized, cattle have to be looked after very well, they have to be housed well, they have to have adequate water, they have to be maintained disease free. Feeding should be also carried out in a scientific manner. Special emphasis on the quality and quantity of the fodder. Stringent cleanliness and hygiene should be required, both of the cattle as well as the handlers, while milking, storage, and transport. And nowadays, all of these processes have become mechanized. Therefore, this reduces the direct contact of the produce with the handler, and the stringent measures and these hygiene. It's all very important. Because all of this is mechanized, therefore regular inspections will be required with proper record keeping, and it would also help to identify and rectify the problems as early as possible. And regular visits by veterinary doctor are also mandatory. Now you would find it interesting that if you, okay, so what are the important points in dairy farm management? Dairying is the management of animals for milk and milk products for human consumption. So dairying is the management of for milk and its products for human consumption. What are the important points that you should remember? Um that is dairying, that is in dairy farm management, we deal with processes and systems which increase the yield and improve the quality of milk, and increasing the yield and improved quality directly depends upon the quality of the breeds. That is good breed selection gives us very high yielding potential along with good breeds. That is, if you get a good breed, then only because you have a good breed does not mean it will increase your yield and quality of milk. You have to take proper care of it. You have to give that is feed, fodder in a scientific manner. That is, proper food and adequate amount of water should be given to the cattle, as well as you should have stringent hygienic conditions, and there should not be any direct contact of produce with the handler. Everything should be mechanized, and if everything is mechanized, then there should be proper inspections, and veterinary doctors should be available, and regular checkups should be there, as well as disease resistance, that is combined with disease resistance, that is good quality of breeds, as well as you should make sure that they do not, that is they stay disease resistant, and proper hygienic care and good care, well housed. All of these are important factors. Then poultry farm management we had studied, that is poultry is the class of domesticated fowl birds used for food or for their eggs. So it is the class of domesticated fowl birds, that is your hen, chicken, etc. And it is used for either themselves, that is the food, or for their eggs. So this typically includes chicken and ducks, but also sometimes turkey and geese. Usually it is just chicken and duck, sometimes turkey and geese too. Poultry is often used to refer to meat of only these birds, but in more general sense it may refer to meat of other birds too. So in many other countries, that is different meats of different birds are consumed. Selection of disease-free and suitable breeds in proper and safe farm condition, proper food and water, hygiene and health, they are all important in again poultry farm management. So the same thing which was there in dairy farm management, or that is the same all factors apply, that is good hygienic condition, proper care, regular inspection, veterinary doctors should be there. Then there should be good selection of good quality of breed, proper food and water should be given, they should be kept disease free. So uh, all the same things. Then comes animal breeding. Breeding of animal is an important aspect of animal husbandry. Animal breeding aims at increasing the yield of animals and improving the desirable qualities of produce. Breeding of animals is very important aspect of animal husbandry. Why? Because it aims at increasing the yield of animals and improving the desirable qualities of the produce. How do we do that? That is, firstly, what is the meaning of breed? A group of animals related by descent and similar in most characters like general appearance, feature, size, configuration are said to belong to a breed. So a group of animals which are firstly related by descent, and they are similar in most characters like general appearance, feature, size, configuration, etc. are said to be belong to a breed. Now when breeding is between two animals, then when breeding is between two animals of the same breed, then it is called as inbreeding. So whenever we breed two animals of the same breed, then it is called as inbreeding. But when there is a cross between two different breeds, then it is called as outbreeding. So firstly, inbreeding. Inbreeding refers to the mating of more closely related individuals within the same breed for 4 to 6 generations. This is very important. Inbreeding refers to the mating of more closely related individuals within the same breed for 4 to 6 generations. The breeding strategy is as follows. That is, superior males and superior females of the same breed are identified and mated in pairs. Okay, so superior males and superior females are identified and then they are mated in pairs. The progeny obtained are evaluated and superior males and females among them are identified for further mating. In inbreeding, you first see that inbreeding is between that is two individuals of the same breed which have common ancestors for 4 to 6 generations. Then you select the most superior male and female and then you breed them to get offsprings. Then out of these offsprings, again you select the most superior male and female and then you further breed them. And a superior female in the case of cattle is the cow or buffalo that produces more milk per lactation. So what is the meaning of superior male or superior female? That is, superior female that is a superior female is in the that is in the case of cattle is the cow or buffalo which produces more milk per lactation. So the cow or buffalo which produces more milk per lactation is called as a superior female. Then on the other hand, the superior male is the bull which gives rise to superior progeny as compared to those of other males. That is the progeny. That is, what is the superior male? That is, superior male is the bull which gives rise to a superior progeny as compared to those of the males. So the progeny from this superior male, all the superior females and superior males would be, so basically more superior progeny will be collected, so there will be produced, so therefore that is called as the superior male. Try to recollect the homozygous pure lines by Mendel. So similar strategy is used to develop pure lines as it was used in the case of P, that is inbreeding increases homozygosity. So inbreeding is necessary if we want to evolve a pure line in any animal. So all those breeds that we get which say that these are pure lines, so you will see that any pure line breed will be having all those diseases and characters and appearance and everything which was there in in from the start of the breed. That is you can say pure line, that is the innate characteristics. So because inbreeding increases homozygosity. So inbreeding exposes harmful recessive genes that are eliminated by selection. So basically through inbreeding all the harmful recessive genes will be exposed, but then we can eliminate them. So it is since it's not, so let's say we got 8 offsprings. Out of those 8 offsprings, that is, since it is pure line, so every time that is we breed more and more, like we can see that okay, this is having this parent had a recessive gene, this had a recessive gene, so now this is recessive. So we know that this is completely recessive. So then we are going to eliminate it by selection, and which will, next time for breeding we will choose those which have superior genes, that is which show dominant characteristic or which show desirable characteristics, not just dominant but desirable characteristics. So inbreeding exposes harmful recessive genes that are eliminated by selection. It also helps in accumulation of superior genes and elimination of less desirable genes. So let's say this has disease resistance and this also has disease resistance genes, so both of them combine and we got and that is in these both disease resistance genes was recessive, or let's say it was dominant, whichever case it may be. So when we realize that this is disease resistance, then for further mating we will choose these genes. So there is accumulation of superior genes, there is accumulation of less desirable gene, or accumulation of more desirable gene, accumulation of less desirable gene, because we are mating between pure lines. So after this, we can select whichever gene we feel is more desirable. We can select them for further mating. Whichever we feel is not desirable, then we can just eliminate them. That is, not mate them further. Therefore, this approach where the selection at each step increases the productivity of inbred population. In this approach, selection at each step increases the productivity. That is, in inbreeding, the productivity increases, because every time you get offsprings, you can find out which are the superior ones, and then further you can inbreed them. So if you inbreed them, then they don't have the recessive gene. So their progeny is always going to have the genes that their parents have. If their parents do not have the less desirable genes and they only have the more desirable genes, then the offspring are also going to have more desirable genes. And every time, in case the recessive gene is expressed, then you can eliminate them, or you can not further mate them, so as to that is prevent that recessive gene from expressing further in the species, or in the mating, that is in the further progeny. But however, continued inbreeding, especially close inbreeding, reduces fertility and even productivity, and this is called as inbreeding depression. So this was there in sexual reproduction of plants also. When continuous autogamy kept happening, that is especially in clistogamous flowers, then it led to inbreeding depression, and eventually that is it leads to reduced fertility, reduced productivity, and eventually it leads to the extinction of species. That is inbreeding depression. Whenever this becomes a problem, selected animals of the breeding population should be mated with unrelated superior animals of the same breed. This usually helps to restore the fertility. What is the meaning of unrelated superior animals of the same breed? It means outcrossing, which we are going to learn further. That is the mating between two individuals of the same breed, but not having any common ancestors for 4 to 6 generations. Above, inbreeding was, that is breeding between two organisms or individuals of the same breed, but they will be having common ancestors for 4 to 6 generations, whereas in this case they will not be having that is unrelated superior animals. They won't be having any common ancestors for 4 to 6 generations. Okay, so now we come to outbreeding. Outbreeding is the breeding of the unrelated animals, which may be between animals of the same breed but having no common ancestors for 4 to 6 generation, which is what, like I just said, outcrossing, or between different breeds, which is crossbreeding, or different species, that is interspecific hybridization. So outbreeding is of 3 types. First is outcrossing, in which there are two individuals which are of the same breed but they have no common ancestors for 4 to 6 generations. That is called as outcrossing. Then secondly, it is that is crossbreeding. What is crossbreeding? That is that is breeding of two individuals of different breed. And then lastly, it is interspecific hybridization, in which there are two individuals of different species species altogether. Outcrossing is the practice of mating animals within the same breed but having no common ancestors on either side of their pedigree up to 4 to 6 generations. Offspring of such a mating is known as an outcross. And it is the best breeding method for animals that are below average in productivity in milk production, growth rate in beef cattle, etc. A single outcross often helps to overcome inbreeding depression. This is a very important line, and there are questions in NEET regarding this. That is, how can we stop inbreeding depression, or inbreeding depression can be stopped by a single outcross. Then after outcrossing, there is crossbreeding. Crossbreeding, in this method, superior males of one breed are mated with superior females of another breed. Crossbreeding, that is superior males of one breed are mated with superior females of another breed. Crossbreeding allows the desirable qualities of two different breeds to be combined. Progeny hybrid animals may themselves be used for commercial production, or they may be subjected to some form of inbreeding to develop new stable breeds. So in crossbreeding, there are breeding of two animals of different breeds. So crossbreeding allows the desirable qualities of two different breeds to be combined, and the progeny of the hybrid animals can themselves be used for commercial production, or they may be subjected to some form of inbreeding, and then they can develop superior new stable breeds that may be superior to the existing breed. Many new animal breeds have been developed by this approach. Hizardale is a new breed of sheep developed in Punjab by crossing between Bikaneri Ewees and Merino Rams. So by crossbreeding, that is superior males of one breed are mated with superior females of another breed, and crossbreeding allows the desirable qualities of two different breeds to be combined. Progeny of hybrid animals may themselves be used for commercial production, or they may be subjected to some form of inbreeding and selection to develop new stable breed which may be superior to the existing breeds. So either you can commercially, that is, you can choose two different breeds with two different desirable characteristics, and you can breed them, or you can, after breeding them, you can inbreed them. That is, you can use them for commercial production, or you can inbreed crossbreed, and you can create a new stable breed which may be superior to the existing breeds. So what are the new animal breeds that have been developed? That is, Hizardale is a new breed of sheep which was developed in Punjab by crossbreeding the Bikaner Ewees and Merino Rams. Now, interspecific hybridization, again, a male and female of two different related species. That is, male and female of two different but related species. Interspecific hybridization is related species are mated. The progeny may combine desirable features of both the parents and may be of considerable economic value. So mule, example mule, that is donkey and horse. Two different related species, male and female animals of two different related species may combine, and they may be of considerable economic value. Example is the mule. Then controlled breeding experiments carried out using artificial insemination. The semen is collected from the male that is chosen as a parent and injected into the reproductive tract of the selected female by the breeder. So basically, artificial insemination takes place, that is controlled breeding experiments. Semen is collected from the male which is chosen as parent and then injected into the reproductive tract of the selected female by the breeder, and it can be used immediately, that is within 4 to 6 hours, or it can be frozen and used at a later date, frozen in nitrogen. It can also be transported in a frozen form where the female is housed. In this way, desirable mating are carried out. Artificial insemination helps us to overcome several problems of normal matings. Now, often success rate of crossing mature male and female is fairly low, even though artificial insemination is carried out. For this MOAT, multiple ovulation embryo transfer technology, is used. This program is for herd improvement. What happens in this? The cow is administered with FSH-like activity hormones, so hormones with FSH-like activity, and these induce follicular maturation and super ovulation. So instead of only one egg, there is which normally they yield per cycle. A cow yields 6 to 8 eggs. So since it yields 6 to 8 eggs, it is either mated with an elite bull or it is artificially inseminated, and then at 8 to 32 cell stage, all these eggs are recovered non-surgically and they are transported to surrogate mothers. So one egg each. So these 8 eggs are transported to, let's say, 8 surrogate mothers, and they are recovered from the superovulated, that is superovulation took place in the cow, so the superior female cow, and they are recovered non-surgically, and then the genetic mother is available for another round of superovulation. So this technology, multiple MOAT, that is multiple ovulation embryo transfer technology, has been demonstrated for cattle, sheep, rabbit, buffalo, mares, etc. High milk yielding breeds of females and high quality lean meat, less lipid milk yielding bulls have been bred successfully to increase herd. So this is also a very important line, that is high milk yielding breeds of females and high quality, that is lean meat with less lipid meat yielding bulls have been bred successfully to increase herd size in a very short time. So in this way you don't have to, uh, you don't have to sorry so in this way you don't have to breed all the cows and buffaloes and bulls in the traditional or conventional way, that is you can choose superior male and female, and in this way you can choose superior male and female, and then you can that is by super ovulation you can produce 6 to 8 eggs, and you can put in the other female cows, and then you will get all the progeny will be desirable progeny. Okay, now beekeeping. Beekeeping is also called as apiculture. It is the maintenance of hives of honeybees for the production of honey. It has been an age-old cottage industry, and honey is a food of high nutritive value. It also finds a use in indigenous systems of medicine. It also produces beeswax. That is, honeybee also produce beeswax, which finds many uses in industry such as preparation of cosmetics and polishes of various kinds. So the increased demand of honey has led to large scale. So beekeeping is also called as apiculture. It is the maintenance of hives of honeybees for the production of honey. It has been an age-old cottage industry. Honey is a food which has very high nutritive value, as well as that is, it is very good for body. It is very healthy, and it has indigenous systems of medicine also. So there is a medicine called as honey tusk, and then there are many other medicine. It is good for your throat and all also, as well as your gut. So honeybee also produces beeswax. Along with honey, that is, honeybee also produces beeswax, which finds many uses in industry such as the preparation of cosmetics, polishes, etc. And then the increased demand of honey has led to large scale beekeeping practices. It has become an established income generating industry, whether practiced on a small or in a large scale. So the increased demand of honey has led to large-scale beekeeping practices, and it has become an established income-generating industry, whether practiced on a small or in a large scale. Beekeeping can be practiced in any area where there are sufficient bee pastures of some wild shrubs, fruit orchards, and cultivated crops. There are several species of honeybees which can be reared. The most common species, especially the one which is found in India very easily, is called as Apis indica. Beehives can be kept in one's courtyard, veranda of the house, or even in the roof. And beekeeping is not labor intensive. So, which is not labor intensive? Beekeeping. It is not at all labor intensive. It can be kept very relatively easy. It does require some specialized knowledge. There are several organizations which teach beekeeping. What are the knowledge or the following points for successful beekeeping? That is knowledge and nature of the habits of honeybees, then selection of suitable location for the keeping of beehives, then catching and hiving of swarms or groups of bees, and management of beehives during different seasons, and handling the collection of honey and beeswax. Bees are pollinators of many of our crop species such as sunflower, brassica, apple and pear. Sunflower, brassica, apple and pear. And keeping hives in crop fields during flowering period increases the pollination efficiency, therefore it will improve the yield, and this improving in yield will be beneficial both from the point of view of the crop yield as well as honey yield. So this is very important. So in short, beekeeping is also called as apiculture. It is the maintenance of hives of honeybees for the production of honey. It is an age-old cottage industry. Honey has a very high nutritive value, as well as it is used in indigenous system of medicine. Honeybees also produce beeswax, which is used in the preparation of cosmetics as well as polishes. Then the demand of honey has led to large-scale beekeeping practices, and it has become an established income-gathering industry. Whether practiced in small or large scale, it does not matter. Then it can be practiced in any area wherein there are sufficient bee pastures of some wild shrub, fruit orchard, cultivated crops. And most common species of bee is Apis indica. It can be kept in one's courtyard, veranda, or roof. It is not labor. Beekeeping is not labor intensive, and there are several organizations that teach beekeeping. But beekeeping is very relatively easy. What you should be knowing, you should be knowing the nature as well as the habits of bees. There should be selection of suitable location for the keeping of beehives. Catching and hiving should be known. Management of beehives during different seasons should also be known. And last but not the least, handling and collection of honey as well as beeswax. Bees are pollinators of sunflower, brassica, apple and pear. So therefore keeping them in crop fields will increase the pollinating efficiency. Therefore it will increase the yield or improve the yield, that is the crop yield as well as your honey yield. Then comes fisheries. Fisheries is an industry devoted to the catching, processing and selling of fish, shellfish and other aquatic animals. That is pisciculture is specially for fishes, and aquaculture is for other aquatic animals. A large number of our population is dependent on fish products and other such as prawn, crab, lobster, edible oyster, etc. These are delicacies that a lot of people enjoy. Seafood, or as you like to say. What are the freshwater fishes? There are three freshwater fishes: katla, rohu and common carp. Katla, rohu and common carp. What are the marine fishes? They are pomfret, hilsa, sardines and mackerel. Hilsa, sardine, mackerel and pomfret. Fisheries have an important place in Indian economy. They provide income and employment to millions of fishermen and farmers, particularly in coastal states, that is in the Western Ghats in Maharashtra, Gujarat and all. And then it is the only source of their livelihood for many people. In order to meet the increasing demands of fisheries, different techniques have been employed to increase production. Through aquaculture and pisciculture, we have been able to increase the production of aquatic plants and animals, both freshwater and marine. This is also very important. That is, increased production of freshwater as well as marine animals has been increased through aquaculture and pisciculture, and this has led to the development of flourishing of fishery industry. It has brought a lot of income to the farmers in particular and country in general. So now we talk about blue revolution, just like we used to talk about green revolution. So during independence we talked about green revolution, wherein the population increased tenfold, and the economy had to also focus on the increase in food production to meet the growing demands of our country, and therefore there were new plant hybridization techniques for crops like wheat, maize, etc. were introduced in particularly the northern states, that is Punjab, Rajasthan, etc. And in these Punjab, Haryana also, I guess, and in these states and in these states, that is these new hybridization techniques, they increase the yield of wheat and maize, and due to this increase, and that is increased food production took place, and this was called as the Green Revolution. Similarly, for for fishes, it is the Blue Revolution. So fisheries devoted to catching, processing, selling. Then increasing different techniques have been employed to increase production. Through aquaculture and pisciculture, we have increased the production of aquatic plants and animals, both freshwater and marine. Development and flourishing of fishery industry, it has brought a lot of income to the farmers. What? Blue Revolution. Now we come to plant breeding. Traditional farming can only yield a limited biomass as food for humans and animals. Better management practices and increase in acreage can increase yield, but only to a limited extent. Plant breeding as a technology has helped increase yields to a very large extent. Who in India has not heard about the Green Revolution, which is responsible for our country to not merely meet the national requirements, but also helped us to even export it. Green Revolution was dependent to a large extent on plant breeding techniques for development of high yielding and disease resistant varieties in wheat, rice and maize. So Green Revolution was dependent to a large extent on plant breeding techniques for development of high yielding and disease resistant varieties, that is in wheat, rice and maize. Now what is plant breeding? Plant breeding is a purposeful manipulation of plant species in order to create desired plant types which are better suited for cultivation and give better yield, and they are disease resistant. So what is plant breeding? Plant breeding is purposeful manipulation, that is intentionally or purposefully we are manipulating plant species in order to create desired plant types which are better suited for cultivation, that is give us more yield or better yield, and they are disease resistant, that is they don't catch diseases so fast and die. So therefore their lifespan also increases. Conventional plant breeding has been practiced for thousands of years since the beginning of human civilization. Recorded evidence of plant breeding dates back to 9,000 to 11,000 years ago. So conventional plant breeding has been practiced for many years. From when do we know? That is by evidence, recorded evidence, that is from 9,000 to 11,000 years ago. From then on, plant breeding has been practiced. Many present day crops are the result of domestication in ancient times. Today, all our major food products are derived from domesticated varieties. Classical plant breeding involves crossing or hybridization of pure lines followed by artificial selection to produce plants which with desirable traits of higher yield, nutrition and resistant to diseases. So classical plant breeding involves the crossing or hybridization of pure lines. So you do crossing or hybridization of pure lines followed by artificial selection. You select and then you produce plants with desirable traits of higher yield, nutrition and resistance to diseases. But with advancement in genetics, molecular biology and tissue culture, plant breeding is now increasingly carried out by using molecular genetic tools. So earlier you used to take two pure lines and then that is like we saw before also, that is in inbreeding, that is you take two pure lines, then you get all the offsprings, okay, the progeny, and then out of those you that is the recessive genes will be exposed, and then artificially you select the ones which are superior and then you further produce them to get your desirable traits, and that is such as increasing in yield, more nutrition and disease resistant. This was classical plant breeding. If we were to list the traits or characters that the breeders have tried to incorporate into plants, first we would list would be increased crop yield and improved quality. This is very important. So firstly, disease resistance and tolerance to environmental stresses and all will come later on. Firstly, if we were to list the characters which breeders have tried to incorporate into crop plants, then we would list increased crop yield, that is to increase the crop yield and to improve the quality. Increased tolerance to environmental stresses such as salinity, that is more saline conditions, that is more salt, that is in sea and all. So extreme temperatures, drought, resistance to pathogens such as virus, fungi, bacteria, and increased tolerance to insect pests would be on our list too. So So increased tolerance to okay. So these would be the characters. So what are the main points? That is, plant breeding is the purposeful or intentional manipulation of plant species in order to create desired plant types which are better suited for cultivation. They give better yield and they are disease resistant. Conventional plant breeding has been practiced for thousands of years, and the recorded evidence of plant breeding dates back to 9,000 to 11,000 years ago. Uh, a lot of crops today are the result of domestication in ancient times. What does classical plant breeding involve? Classical plant breeding involves a crossing or hybridization of pure lines, and then artificial selection takes place to produce the plants with desirable traits which give us higher yield, nutrition, and resistance to diseases. With advancement in genetics nowadays, we do not practice conventional and classical plant breeding. Rather, we plant breeding or hybridization is done more, that is with molecular genetic tools. If we were to list the traits or characters, that is firstly the character that the breeders would like to incorporate in plants would be increased crop yield. Just a second. So if we were to if we were to if we were to list the traits that the breeders have tried to if we were to list the traits that the breeders have tried to put, then firstly it would be increased crop yield and improved quality. Then secondly it would be increased tolerance to the environmental stresses, salinity, extreme temperature, drought, resistance to pathogen, virus, fungi, bacteria, etc., and increased tolerance to insect pests. So, but firstly it would be crop yield and improved quality. Now, plant breeding programs are carried out in a systematic way worldwide in government institutions and commercial companies. The main steps in breeding a new genetic variety of crop are collection of variability, so firstly collection of variability. So how to that is how do we breed a new genetic variety? First is genetic variability is the root of any breeding program. In many crops pre-existing genetic variability is available from wild relatives of the crop. Collection and preservation of all different wild varieties, species and relatives of the cultivated species followed by their evaluation is a prerequisite for effective exploitation. Okay, so for any breeding program, what is the most important thing? For any breeding program, the most important thing is genetic variability. So, all so if we want that all our further progeny has to be different, that is has to be disease resistant. Let's say there is a plant that is its progeny for many generations. It has been producing seeds and the plants that is its further progeny for many generations, they have all been diseased. That is after a certain, that is after 1 or 2 years, that is all of them, they are diseased or they die very fast. That is as soon as we plant it, as soon as they are grown, or there is some kind of, that is it is continuously producing small leaves or small fruits or less sweet fruits or something like that, and what do we need now? We need variation, that is we need variability. We want that in the next generation of this plant, that is the progeny, will not be having such kind of characteristics, that is these less desirable characteristics. So in many crops pre-existing genetic variability is available from wild relatives of the crops. So let's say I have a rose plant which does not flower very properly, or does not have proper, that is has lesser small leaves or something, but I got another rose plant of the same variety, same species, everything is same, it's just that it is growing very nicely, or there are, that is after breeding two rose plants, that is the rose plant that I got, it has characteristics which are desirable, whereas these both did not have, or this has less desirable characteristic, whereas one of these parents has the desirable characteristic that I want. So in many crops, pre-existing genetic variability is available from wild relatives of the crop. Collection and preservation of all the different wild varieties. So I take all the varieties that I like, that is having the desirable characters, I collect them and then I preserve them, and species and relatives of the cultivated species, followed by their evaluation for the characteristics. Then I evaluate them, that okay, this is the characteristic, disease resistance I want, okay, bigger leaves I want, okay, yeah, sweetness in fruit I need, that is pest and insect resistance also I need. So I select them, I evaluate them, and is a prerequisite for effective exploitation of natural genes available in the population. So these are all the natural genes which were there in the plant, in the wild relatives of the plant, or that is it was there basically in the species, it's just that now I found it. So now I will exploit these natural genes. The entire collection of plant seeds having all the diverse alleles for all genes in a crop is called as germplasm collection. So all of these, the entire collection of plants or seeds having all the diverse alleles for all the genes in a given crop is called as germplasm collection. Now evaluation and selection of parents, that is the germplasm, is then evaluated so as to identify plants with desirable combination of characters. Now we found the germplasm, now it will be evaluated so as to identify the plants with desirable combination of characters. So now I need, that is the male should be having disease resistance and better color of plants, or the female would be having like bigger leaves and insect and pest resistance. So now we combine the characters, desirable combination of the characters. The selected plants are multiplied and used in the process of hybridization. Pure lines are created wherever desirable and possible. So now we took this, and now we multiplied, and that is hybridization took place, and pure lines will be created wherever desirable and possible. Now cross hybridization among the selected parents. The desired characters are very often to be combined from two different plants or parents. For example, high protein quality in one parent may need to be combined with disease resistance from another parent. So now let's say I selected from two different plants, so combined all the characters, and this has now very high protein quality and this has disease resistance. Okay, this is disease resistance, and this is possible by cross hybridizing the two parents to produce hybrids that genetically combine the desired characters in one plant. So this is possible by cross hybridizing the two parents. This is possible by cross hybridizing the two parents to produce hybrids that genetically combine the desired characters in one plant. So now cross hybridization in one plant, I got both, that is high protein quality as well as disease resistance. This is a very time consuming and tedious process, since pollen grain from the desired plant chosen as male have to be collected and placed on the stigma of the flower selected as female, and we know how to do this, right? So if it is a bisexual flower, then emasculation will take place, and then like if it is a stigma, then bagging has to be done, and then when the stigma will get receptive, then we will finally collect the pollen grains, and then we will like all those things that if this is a bisexual flower, this is the stigma, this is the stamen, then firstly we need stamen from another desired plant that we saw, that is who has higher protein quality. We know this has disease resistance, so first we will do emasculation, we will remove the male, then we will bag the female, that is the stigma, then we will wait the stigma to be receptive, then we will collect the pollen over here. If the pollen becomes receptive first, then we will collect the pollen grains and we will store it in liquid nitrogen till the stigma gets receptive, so that it does not that is, it does not go bad. Then we will transfer whenever the stigma gets receptive, then we will transfer the pollen grains over here, and hybridization will take place. So also it is not necessary that hybrids do combine the desirable characters. Usually 1 in few hundred to thousand crosses shows the desirable combination. So there was an example wherein the scientist wanted, that is tomato plant above, and I think potato or carrot or something like that below, that is root and stem modification. So dahlia, I guess. So root and stem modification, I think they wanted, and the moment they crossed it, they found a plant which that is which had the stem of, I think, dahlia and the root of potato or something like that. So basically, in a few, that is only few hundred to 1,000 crosses show the desirable combination that we want. Now, selection and testing of superior recombinants. That is, this step consists of selecting among the progeny of hybrids those plants that have the desired character combination. The selection process is crucial to the success of the breeding objective and requires careful scientific evaluation of the progeny. Selection and testing of superior recombinants. That is, this step consists of selecting among the progeny of the hybrids those plants that have the desired character combination. So now we produce the hybrids. Now we see that now all the hybrids are not going to have desirable combination, right? Because only few hundred in 1,000 crosses show the desirable combination. So in this case, firstly we will select among the progeny of hybrids which have the desired character combination. After the selection process is done, because success of a breeding objective, that is, it is crucial. The selection process is very crucial to the success of the breeding objective. Our objective was to get tomato as well as, let's say, sweet potato, right? So because sweet potato is a stem modification. So it is, so after that is, it is crucial to success of the breeding objective. It requires careful scientific evaluation of the progeny. This step yields plants that are superior to both of the parents. Very often more than one superior progeny may become available. These are self-pollinated for several generations till they reach a state of uniformity or homozygosity, so that the characters will not segregate in the progeny. So this step yields plants that are superior to both of the parents. Very often one is more superior progeny may become available. Then after we find this, that is, we take these two superior ones that we got hybrid, and then we self-pollinate. That is, we either self-pollinate them, or if we found two, then that is basically we need to maintain uniform homozygosity so that the characters will not segregate in the progeny. Till we produce them, self-pollinate them for several generations, till they reach a state of uniformity or homozygosity, that is pure lines basically. That is, we created a new breed of it, right? Now, testing, release and commercialization of new cultivars. The newly selected lines are evaluated for their yield and other agronomic traits of quality, disease resistance, etc. The evaluation is done by growing these in the research fields and recording their performance under ideal fertilizer application, irrigation and other crop management practices. So testing, release and commercialization. So newly selected lines, that we produce the newly selected line, right? So first, then these are evaluated for yield and other agronomic traits of quality, disease resistance, etc. So now is the entire line, that is, are these actually like agronomically that is useful? That is, do they actually increase the yield, or do they actually are disease resistant? So we evaluate and then record their performance under ideal fertilizer application, irrigation and other crop management practices. Then evaluation in the research field is followed by testing the material in the farmer's field. So first, researchers field we test, then in the farmer's field for at least 3 growing seasons at several locations in the country. In the farmer's field, the testing takes place, and then representing all the agroclimatic zones where the crop is usually grown, material is evaluated in comparison to the best available local crop cultivar, a check of the reference cultivar. So after this, the material is evaluated in comparison. That is, if we, let's say, we founded a new variety of rice, so then now we are going to that is first from the original variety of rice from wild relatives, we took the ones which were like desirable characters. Then we then we produced the hybrid. Then again we selected and evaluated, that is the one which has the desirable characters. Then we that is then after we found the hybrid, we for many generations we self-pollinated it, and we made like a homozygosity, maintained the homozygosity pure line. Then in the researchers field we tried it. Then in the farmers field for at least 3 growing seasons in different parts of the country, different agroclimatic zones, we checked whether it actually has those desirable characters such as increase in yield or disease resistance or all those characters that we want. and after that we compare it with our previous species of variety of rice that we had or we had taken, and after that we make it available at a local crop cultivar. Then India is mainly an agricultural country. Agriculture accounts for 33% of India's GDP, that is gross domestic product, and employs nearly 62% of the population. This is a huge number. 62% of the population of India is being employed in agriculture sector, and 33% of India's GDP agriculture accounts for. After India's independence, one of the main challenges facing the country was that of producing enough food for the increasing population. So only limited this is what I said above before also. That is, after India's independence, the main challenge was that the population became very high, and the country had to produce enough food for the increasing population. As only limited land was fit for cultivation, India had to strive to increase yields per unit area for existing farmland. That is, all that farmland that we had, in that farmland only we had to, that is, quadruple or double the yield that we got per unit area in a particular time. So development of several high-yielding varieties of wheat and rice in the mid-1960s. As a result, various plant breeding techniques led to dramatic increase in food production, and this phase is often referred to as Green Revolution. During, that is, during the time wherein, you know, in many parts of the Europe and all, you must have read in previous classes, there was shortage of food and all. In our country, due to Green Revolution, that is, it is a very huge big success. That is, high yielding varieties of wheat and rice in 1960 led to various plant breeding techniques, and it increased the food production in our country, because of which हमारा हाल बाकी लोगों की तरह नहीं हुआ. Therefore, this is called as Green Revolution. Now, before going over there, let's just quickly, I'll revise what we just read. So, first collection of variability, that is genetic variability is obviously the root of any breeding program, and pre-existing genetic variability was found in wild relatives of the crop. Therefore, collection and preservation of all different wild varieties, species and relatives of the cultivated species, followed by the evaluation of the characteristics, was done for effective exploitation of the natural genes. In this, we are exploiting the natural gene, and the entire collection of the plants or genes which had all the diverse alleles for all the genes in a given crop was called as germplasm collection. Then evaluation and selection of the parents took place. That is, it was evaluated so as to identify plants with desirable combination of characters. Then they were multiplied and used in the process of hybridization, and pure lines were created wherever possible. Then cross hybridization occurred among the selected parents. That is, the desired characters are very often combined from two different plants or parents. That is, high protein for one and disease resistant for other. And then this was possible by cross hybridizing to produce hybrids which genetically combine the characters in one plant. Then this was very time consuming and tedious, because tedious. Why? Because we had to take the pollen grain from the desirable and then put it in the sigma and all. And after this, it was not necessary that the hybrids combined the desirable characters, because only a few hundred in 1,000 crosses showed the desirable characteristics that we wanted. After that, selection and testing, that is among the progeny of the hybrid, to have desired character combination. It was crucial to the success of the breeding objective and required careful scientific evaluation of the progeny. This step yields plants are superior to both the parents. Very often one may be superior. After that, they were self-pollinated for several generations till they reached a state of homozygosity or uniformity, so that they won't segregate in the progeny. These desirable characteristics, they had to reach a uniformity or homozygosity, that is in each generation both the plant had to have all these characteristics every time, and they all had to be dominant, so that these characteristics would reach a uniformity or homozygosity and they won't get segregated. If they got segregated, then there was no meaning of doing itna sab hybridization and all. So after this, the new commercialization and new testing was done, and these selected lines were evaluated for their yield and other agronomic traits, and research fees according to their performance, segregation and other crop management. And this research was followed by testing it. So first in research field it was done, and then in the farmer field for 3 growing seasons at several locations of the country. This testing was done, and after where the agroclimatic zones where the crop is usually grown, and then it was compared to the best available local crop cultivar to check or reference, and then it was sold in the market. Okay. Now, in India, India is an agricultural country, we all know that. Then agriculture accounts for 33% of India's GDP. It employs 62% of the population. These are very important lines. And what happened in Green Revolution, that is after India's independence, one of the main challenges was that is the production of food. Only limited land was fit for cultivation in India. Therefore, to increase the yield per unit area from existing farmland, the development of several high-yielding varieties of wheat and rice in the mid-1960s as a result of various plant breeding techniques led to dramatic increase in food production in our country, and this phase was referred to as Green Revolution. Now we come to wheat and rice. Wheat and rice. During the period 1960 to 2000, wheat production increased from 11 million tons to 75 million tons. This is very important. That is, during the period of 1960 to 2000, wheat production from 11 million tons due to Green Revolution from 11 million tons to 75 million tons. While rice production, this is for wheat production, rice production went from 35 million to 89.5 million tons. Can you believe that? This was due to development of semi-dwarf varieties of wheat and rice. Why was this? This was due to the development of semi-dwarf varieties of wheat and rice. Nobel laureate Norman E. Borlaug at International Center of Wheat and Maize Improvement in Mexico. These are all very important. International Center for Wheat and Maize Improvement, Mexico, developed the semi-dwarf. These are very important lines. So this period, this entire part is actually important. Okay, so that is wheat production increased from 7 million tons. Wheat production increased from 7 million tons to 75 million tons in a period of 1960 to 2000, and the rice increased from 35 million tons to 89.5 million tons in 1960 to 2000, and this was due to the development of semi-dwarf varieties of wheat and rice. And Nobel laureate Norman E. Borlaug at the International Center for Wheat and Maize Improvement in Mexico developed the semi-dwarf wheat. In 1963, several varieties such as Sonalika and Kalyan Sona, which were high yielding and disease resistant, were introduced all over the wheat growing belt of India. That is, several varieties such as Sonalika and Kalyan Sona, which were high yielding and disease resistant, were introduced all over the wheat growing belt in India. Semi-dwarf rice varieties were developed from IR8, that is International Rice Research Institute, Philippines. Where is the International Rice Research Institute, Philippines? And Taichung Native 1 in Taiwan. Taichung Native 1 in Taiwan. These were the semi-dwarf varieties, and they were derived from here. They were introduced in 1966, but better yielding semi-dwarf varieties, Jaya and Ratna, which were developed in India, were then made, and they were circulated. So, what all is there? That is, firstly, during the period of 1960 to 2000, the wheat production went from 11 million tons to 75 million tons, while the rice production went from 35 million tons to 89.5 million tons. Why was this? This was due to the development of semi-dwarf varieties of wheat and rice. Who was the Nobel laureate? Nobel laureate Norman E. Borlaug at International Center for Wheat and Maize Improvement in Mexico. He developed the semi-dwarf variety of wheat in 1963, and several varieties such as Sonalika and Kalyan Sona, they were high yielding and disease resistant, were introduced all over the wheat growing belt in India. So obviously, firstly, Norman E. Borlaug was a Nobel laureate. He developed it, but then later on, Sonalika and Kalyan Sona were introduced in India, and they were high yielding and disease resistant, and all over the wheat growing belt in India. Semi-dwarf variety of rice were first derived from IR8, which was developed in International Rice Research Institute (IRRI) in Philippines, and Taichung Native 1, which was in Taiwan. But the derivatives were introduced in 1966, but later on, better yielding semi-dwarf varieties, Jaya and Ratna, were developed in India itself. Now sugarcane. Sugarcane, Saccharum berberi was originally grown in North India, but it had poor sugar content and yield. Tropical canes grown in South India, Saccharum officinarum. So Saccharum barberry was originally grown in North India, but it had very poor sugar content and yield. And tropical canes grown in South India, such as Saccharum opinarum, had thicker stems and it had higher sugar content, but it did not grow well in North India. These two species were successfully crossed to get sugarcane varieties combining the desirable qualities of high yield, thick stem, high sugar, and ability to grow in the sugarcane areas of North India. So Saccharum barberry was originally grown in North India, but it did not grow very well. That is, the yield was also poor and sugar content was also poor. Tropical canes, which was grown in South India, which was Saccharum opinarum. Saccharum opinarum had thicker stem and it had high sugar content, but it did not grow well in North India. So these two species were successfully crossed to get sugarcane varieties which combine the desirable qualities, that is high yield भी था, plus thick stem भी था, plus high sugar भी था, plus not maybe it grew. Then millets. Hybrid maize, jowar, bajra have been successfully developed in India. Hybrid breeding have led to the development of several high-yielding varieties resistant to water stress. So in millets, hybrid maize, jowar, hybrid jowar and hybrid bajra have been successfully developed in India. Hybrid breeding have led to the development of several high-yielding varieties which are resistant to water stress. Now, plant breeding for disease resistance. So Saccharum barberry was originally grown in North India, but it had poor sugar content as well as poor yield. Tropical cane, which was grown in South India, was Saccharum officinarum, which had thicker stem and high sugar content, but it did not grow well in North India. So there was a crossing or hybridization between both of these species, and it combined the desirable qualities, that is high yield, thick stem, high sugar content, and ability to grow in areas of North India as well. Millets, hybrid maize, hybrid jowar and hybrid bajra have been successfully developed in India, and they have led to the development of that is several high yielding varieties which are resistant to water stress. Now, plant breeding for disease resistance. A wide range of fungal, bacterial and viral pathogens. That is, a wide range of fungal, bacterial and viral pathogens affect the yield of cultivated crop species, especially in tropical climates. Crop losses can often be significant, up to 20 to 30%, or sometimes even total. Now we come to plant breeding for disease resistance. So there are a wide range of fungal as well as bacterial as well as viral pathogens, that is disease causing organisms. Which affect the yield of cultivated crop species, especially in tropical climate. That is, they affect the yield of cultivated crop species, especially in tropical climates. Crop losses can often be significant, up to 20 to 30%, or sometimes even total. So there are a wide range of fungal, bacterial and viral pathogens which affect the yield of the cultivated crop species, especially in tropical climates, and the crop losses can often be significant, up to 20 to 30%, or even total. So because of these fungal, bacterial and viral pathogens, that is, the yield of the crop species can be affected, and especially in tropical climates, and we can lose the crop, and the crop loss can be significant, that is 20 to 30%, or even total crop loss can happen. In this situation, breeding and development of cultivar resistant to disease enhances the food production. So it is very necessary for us to now grow disease resistant, that is cultivars, so that our food production can be enhanced. And this also helps to reduce the dependence on the use of fungicides and bactericides, because they are not good, right? That is, we are always advised to wash our fruits and vegetables thoroughly before eating, because there may be many insecticides, pesticides, fungicide, bactericides and all, and they are extremely dangerous to our health. So disease resistant, because they contain chemicals. This, resistance of the host plant is the ability to prevent the pathogen from causing disease and is determined by the genetic constitution of the host plant. So what is the resistance of the host plant? The resistance of the host plant is the ability to prevent the pathogen from causing disease, and it is determined by the genetic constitution of the host plant. Before breeding is undertaken, it is important to know about the causative organism and the mode of transmission. So you can't just breed that, okay, disease resistant plant, this will be disease resistant, let's do it. Disease resistant to what? That is, to which organism, and what happens in that disease. So the mode of transmission of the disease, how it is transmitted from one plant to another, all of these have to be known. Otherwise you can't breed disease resistant varieties of plants. Some of these diseases which are caused by fungi are rust, that is rust of wheat. We have commonly heard, that is brown rust of wheat, then red rot of sugarcane, and late blight of potato. So what are the diseases caused in plants by fungi? Three examples, that is brown rust of wheat, red rot of sugarcane, and late blight of potato. Then by bacteria there is only one, that is black rot of crucifers, and by viruses there is the tobacco mosaic virus and the turnip mosaic virus. So that is fungi. There are three diseases. Then bacteria, there is one disease given in the NCERT, and virus there are again two diseases. Fungi, it is brown rust of wheat, and late blight of potato, that is brown rust of wheat, then late blight of potato, and red rot of sugarcane. Then bacteria, there is black rot of crucifers, and viruses, there is tobacco mosaic virus and turnip mosaic virus. Methods of breeding for disease resistance. Breeding is carried out by conventional breeding techniques described earlier or by mutation breeding. So how is the breeding for disease resistance carried out? It is carried out by either conventional breeding techniques which were described earlier, that is genetically you will see the genes which are desirable, that is for disease resistant, then you will artificially select them, then you will crossbreed them, then you will see the pure lines, and you will eradicate the less desirable ones which are not disease resistant, then you will choose the superior ones, and then you will breed them again, then you will evaluate them, then you will test them, then you will form a line of them, and all those things. Or it can be done by mutation breeding. So now the conventional method of breeding for disease resistance is that hybridization and selection, which we just saw. Its steps are essentially identical to those for breeding for any other agronomic character such as high yield. So all those characters that we saw for high yield, they are the same for disease resistance as well. The various sequential steps are, like we saw, first is screening germplasm, that is from wild relatives and resistant sources, that is we see, then we collect all the gene, all the alleles, that is which is called as germplasm, then we screen it. Okay, then for resistant sources, then we select the plants which are superior and resistant, then hybridization takes place, then hybrids are produced. Out of those hybrids we see that which are desirable and which are not, so selection and evaluation of hybrids, then testing takes place, and then that is in research fields and all, then in farmer field the testing will take place in different parts of the country, in different agronomic conditions and climatic conditions, agronomic characters, different climatic conditions, and then release will take place. So some crop varieties are bred by hybridization and selection for disease resistance to fungi, bacteria, and viral diseases in table 9.1. So wheat, this is very important, all of these are very important, all of these. Wheat variety is Himgiri. It is resistant to leaf and stripe rust and hill bunt. Then Brassica, Pusa swarnim or Karan Rai. It is resistant to white rust. Cauliflower, Pusa shubra and Pusa snowball K1, which is resistant to black rot and curl blight, black rot. Then cowpea, which is resistant to Pusa komal and bacterial blight. Chili, which is resistant to Pusa sadabahar and chili mosaic virus, tobacco mosaic virus, leaf curl. So let's try to remember it. Wheat. So in wheat, which is the variety, that is Himgiri. Himgiri is resistant to what? Leaf and stripe rust, and hill burnt. Then Brassica. Brassica, what are the varieties? Pusa swarnim, which is also called as Karan Rai. It is resistant to white rust. Okay. Then cauliflower, Pusa Shubhra and Pusa Snowball K1, which is resistant to black rot and curl blight black rot. Then cowpea variety is Pusa Komal. It is resistant to bacterial blight. Then crop is chili. Variety is Pusa Sadabahar. It is resistant to chili mosaic virus, tobacco mosaic virus, and leaf curl. Now, conventional breeding is often constrained by the availability of limited number of disease-resistant genes that are present and identified in various crop varieties or wild relatives, inducing mutation in plant through diverse means and then screening the plant material for resistance sometimes leads to desirable genes being identified. So, conventional breeding is often constrained by the availability of limited number of disease-resistant genes. This is the most important part. That is, in one that is plant, even though you take all the wild relatives, you can't find all the disease-resistant genes, right? So there will be a number of disease-resistant genes which are present and identified in various crop varieties or wild relatives. Inducing mutation in plant, however, through diverse means and then screening the plant materials for resistance sometimes leads to desirable genes being identified. So if you produce a mutation and then you do the same germplasm screening and resistance and all those things, then sometimes you can get desirable genes being identified. This is very important. So plants having these desirable characters can then be either multiplied directly or can be used in breeding. Other breeding methods that are used in selection amongst soma clonal variants are genetic engineering. So what are the other breeding methods that are used in selection among soma clonal variants okay, so other are soma clonal variants and genetic engineering. Soma clonal variants and genetic engineering are the other methods, other breeding methods used for selection. Let's revise till here. Then we'll go ahead. So plant breeding for disease resistance. That is, there is a wide range of fungal, bacterial and viral pathogens and they affect the yield of cultivated crop species, especially in tropical climate, and the crop losses can be that is 20 to 30% or sometimes even total. Therefore, breeding and development is for cultivars which are resistant to diseases is very important, and it enhances the food production. It also helps us reduce the dependence on fungicide and bactericide. Resistance of the host plant is the ability to prevent the pathogen from causing disease, determined by the genetic constitution. So before breeding, we have to know all about the causative organism and the mode of transmission. What are the diseases which are caused by fungi are rust, brown rust of wheat, red rot of sugarcane, late blight of potato. Then by bacteria, it is black rot of crucifers. By virus, it is tobacco and turnip mosaic virus. Now methods for breeding, that is, it can be carried out by conventional breeding techniques, that is, or it can be done by mutation breeding. Conventional breeding is that of hybridization and selection, that is, its steps are identical to those for breeding, that is, various steps include that is screening of germplasm, then resistance sources, hybridization of selected parents, selection evaluation, and then finally release. What are the important ones? Okay, so now let's come to the important ones, that is wheat. What is the variety of wheat? Himgiri. What is Himgiri resistant to? That is leaf and stripe rust, and hillbunt. Then second is brassica. That is the crop is brassica. What is the variety of brassica? The variety of brassica is Pusa swarneem, which is also called as Karan Rai, and it is resistant to white rust. Then comes cauliflower. So cauliflower is Pusa Shubhra and Pusa Snowball K1. Black rot and curl blight black rot. So one is black rot, then is curl blight black rot. Then cowpea Pusa Komal, bacterial blight. Chili Pusa Sadabahar. Chili is Pusa Sadabahar, chili mosaic virus, tobacco mosaic virus, and leaf curl. So wheat, the variety is Himgiri, that is, it is resistant to leaf and stripe rust and hillbunt. Then brassica is also called as Pusa swarneem or Karan Rai. It is resistant to white rust. Then cauliflower is Pusa Shubhra and Pusa Snowball K1, which is resistant to black rot and curl blight black rot. Then cowpea is Pusa Komal, which is resistant to bacterial blight. and chili is Pusa sadabahar, which is resistant to chili mosaic virus, tobacco mosaic virus, and leaf curl. Then conventional breeding is often constrained. Why? Because of the availability of limited number of disease-resistant genes which are present and identified in various crop varieties and wild relatives. What happens in inducing mutation, that is in screening of plant material for resistance, sometimes leads to desirable genes when we induce mutation, and plants which have the desirable characters, they can either be multiplied directly or be used in breeding. Other breeding, what are the other breeding methods for selection? That is soma clonal variants and genetic engineering. First we come to mutation. Mutation is a process by which genetic variations are created through changes in the base sequence within genes. Which we have seen in chapter 5. So mutation is a process by which genetic variations can be created, resulting in the creation of new character or trait not found in parental type. So we can change the base sequence. If we change the base sequence, that is A, T, G, C, if this is suddenly this goes away, so it is GC, right? GC goes away. So then it will become A, C, E, G. In this way it will keep going. So what is happening, that is mutation is taking place. So in frame shift, that is if this used to come in a frame, right, these three used to come in a frame. So now over here let's say there is A, T, so this will shift over here. So the frame will only change. So the frame will change, the protein that it would be coding for will also change, because codon, that is 3. So due to one point mutation, that is frame shift mutation, took place. Now it is possible to induce mutation artificially through the use of chemical or radiation like gamma radiation, and selecting and using the plants that have desirable character as a source in breeding. This process is called as mutation breeding. In mung bean, resistance to yellow mosaic virus and powdery mildew were induced by mutations. So what is mutation? Mutation is the process by which genetic variations are created by through changes in the base sequence within the genes, resulting in the creation of new character or trait which was not found in the parental type. It is possible to induce such mutations artificially through the use of chemicals or radiation, for example gamma radiation, and selecting and using the plants which have the desirable character as a source in breeding. What is this process called? This process is called as mutation breeding. And in mung bean, resistance to yellow mosaic virus and powdery mildew are induced by mutation. This process is called as mutation breeding. And in mung bean, the resistance to yellow mosaic virus as well as powdery mildew, they were all induced by mutations. Now several wild relatives of cultivated species of plants have been shown to have certain resistant characters but have very low yield. So several wild relatives of cultivated species of plants have been shown to have certain resistant characters, but they have very low yield. Hence, there is a need to introduce the resistant gene into high-yielding cultivated varieties. Resistance to yellow mosaic virus in bhindi, which is called as Abelmochus esculentus, was transferred from a wild species and resulted in a new variety, Abelmochus esculentus of this species, which is called as Parbhani Kranti. Okay. So there are many wild relatives of different cultivated species which have been shown to which have been seen to show disease-resistant characters, but they have very low yield. Hence, there is a need to introduce a resistant gene into high-yielding cultivated varieties. The resistance to yellow mosaic virus in bhindi was transferred from a wild species to bhindi, and this variety was called as Parbhani Kranti. All the above examples involve sources of resistance genes that are in the same crop species, which has to be bred for disease resistant, or in a related wild species. All the above example involve sources of resistant gene which are in the same crop species. Either the resistance genes are in the same crop species which has to be bred for disease resistance, or in a related wild species. Transfer of resistance gene is achieved by sexual hybridization between target and the source plant, followed by selection. Okay, so now we come to plant breeding for developing resistance to insect pests. So earlier we saw that is the disease resistance against pathogens such as fungi, bacteria, virus. Now we will see insect pests. Okay, another major cause for large-scale destruction of crop, plant and crop produce is insect and pest infestation. Insect resistance in crop plants may be due to morphological, biochemical or physiological characters. So for disease resistance, it was mainly due to genetic constitution, right? It was due to genetic constitution. But over here, the insect resistance in host crop plants can be due to morphological, biochemical or physiological characteristics. That is morphologically. Venus flytrap and all, they so morphologically they are meant to eat such insects and pests. It can be biochemical, it can be physiological also. Hairy leaves in several plants are associated with resistance to insect pests. Resistance to jazids in cotton and cereal leaf beetle in wheat. It is due to hairy leaves. In wheat, solid stems lead to non-preference by the stem sawfly. So in wheat there are solid stems. If there are solid stems, then the stem sawfly will not prefer them. And smooth-leaved and nectarless cotton varieties do not attract bollworms. So if there is smooth-leaved cotton and nectarless cotton, then it will not attract bollworms. High aspartic acid, low nitrogen and sugar content in maize leads to resistance to maize stem borers. So high aspartic acid, low nitrogen and low sugar content in maize leads to resistance to maize stem borers. So when there is very high aspartic acid, low nitrogen and low sugar content in maize, then maize borers will become resistant to it. So breeding methods for insect pest resistance may involve the same steps as those for any other agronomic trait, yield or quality as discussed earlier. The same that is between crop varieties and all, finding the desirable characters which are insect and pest resistant, and the same thing entire that we saw, that is selecting, then evaluating, then testing, crossing and all that. Sources of resistance from gene pool screening and all. Sources of resistant genes may be cultivated varieties, germplasm collection of the crop or wild relatives. So it can be cultivated varieties, it can be germplasm collection of the crop or wild relatives. Some released crop varieties bred by hybridization are in the table 9.2, that is brassica, rapeseed mustard. Pusa gurao is the variety, and insect pest aphids. Aphids came in Arenotoki, Thilotoki. Thilotoki was aphids, right? No, not Thilotoki, Amphitoki. Amphitoki was aphids, as far as I can remember, in reproduction in organisms. That is, Thilotoki was that is parthenogenesis from female to female. Then that is from female to female. Then Rhinotoki was from female to male, which happened in bees, that is drone bees and wasps. And Amphitoki was in aphids, that is both by parthenogenesis, males and females both can be formed. I'll have to check once though, although I remember aphids from there only. Pusa gurao, brassica, rapeseed mustard, insect pests is aphids. Then crop is flat bean, Pusa sem 2, Pusa sem 3, jazids, aphids and fruit borer. Then okra or bhindi, Pusa savani, Pusa A4, shoot and fruit borer. They are resistant to these. Then plant breeding for improved food quality, that is more than 840 million people in the world do not have adequate food to meet their daily food and nutritional requirement. A far greater number, that is 3 billion people, suffer from micronutrient, protein and vitamin deficiencies or hidden hunger because they cannot afford to buy fruit, vegetable, legumes, fish, meat, and diet lacking these essential micronutrients, particularly iron, vitamin A, iodine, zinc, increase the risk for disease, reduce the lifespan and reduce mental abilities. So for this, what is being done? Biofortifications is very important. In fact, this is a separate entire part. If you go ahead and you read, that is, it is a entire different subject only. Biofortification, breeding crops with higher levels of vitamin, mineral, high protein and healthier fat. It is the most practical means to improve public health. That is, breeding crops with higher levels of vitamins and minerals or higher protein and healthier fats. It is the most practical means to improve public health. Breeding for improved nutritional quality is undertaken with the objectives of improving protein content and quality, oil content and quality, vitamin content and micronutrient and mineral content. Biofortification. What is biofortification? It is breeding crops with higher levels of vitamins and minerals. Breeding crops with higher levels of vitamins and minerals or high protein and healthier fats. It is the most practical means to improve public health, and breeding for improved nutritional quality is undertaken with the objective of improving the protein content and quality, the oil content and quality, the vitamin content, and the micronutrient and mineral content. Then in 2000, maize hybrids had twice the amount of amino acids. That is, the maize hybrids which had twice the amount of amino acids, lysine and tryptophan compared to existing maize hybrids were developed, and wheat variety Atlas 66 has a high protein content and has been used as a donor for improving cultivated wheat. It has been possible to develop an iron fortified rice variety containing over 5 times as much iron as in commonly consumed varieties. So all of this you have to remember. There's no way to escape. There's nothing understandable in this. The Indian Agricultural Research Institute of New Delhi has also released several vegetable crops which are rich in vitamins and minerals. Example is vitamin A enriched carrot, spinach, pumpkin, vitamin C enriched bitter gourd, bathua, mustard, tomato, iron and calcium, and enriched spinach and bathua, protein enriched beans, broad, lub, French and garden peas. Okay. Now, single cell protein. The conventional agricultural production of cereal, pulses, vegetables, fruits, etc. may not be able to meet the demand of food at the rate at which human and animal population is increasing. So, the conventional agricultural production of cereal, pulses, vegetables, fruit, etc. may not be able to meet the demand of food at the rate at which human population is increasing. The shift from grain to meat diet also creates a lot more demand for cereals, as it takes 3 to 10 kg of grain to produce 1 kg of meat by animal farming. It's a good point. So, conventional agricultural production of cereal, so the conventionally, like we produce cereals, pulses, vegetable, fruit, etc., it is not sufficient, or it may not be able to meet the demand of the food at the rate at which human and animal population is increasing. So at the rate at which we are increasing, at the rate at which industrialization is taking place and technological improvement, development is taking place, so there are lesser and lesser of the greeneries and areas around us, then it is very difficult that is for us to continue the conventional agricultural production. The shift from grain to meat diet also creates more demand for cereal, because it takes 3 to, that is, the more and more people when they shift to meat diet, then it takes 3 to 10 kgs of grains to that is to produce 1 kg of meat, because you have to buy animal farming, because you have to feed them, right? Can you explain the statement? Yes, because then they'll have to eat that much. So as to get good quality of meat, you need to give them good quality of food. So more than 25%, that is all those people that eat beef, they have to give good fodder to that cow to have good beef. That all those people that eat, that is, you know, that is in America, that is good quality beef and bad quality. So more and more costly will be the better the quality, the more the grain that is the animal has eaten, the better will be the quality of the meat, and the more will be the price of it. So that's how it works. So more than 25% of human population is suffering from hunger and malnutrition. One of the alternate sources of proteins for animal and human nutrition is single cell protein, SCP. So one of the alternate sources of protein for animal and human nutrition is single cell protein, SCP. Microbes are being grown on an industrial scale as source of good protein. Microbes are being grown on an industrial scale as source of good protein. So blue-green algae like spirulina can be grown easily on materials like wastewater from potato processing plants containing starch, straw, molasses, animal manure, even sewage to produce large quantities, and can serve as food rich in protein, minerals, fats, carbohydrates, and vitamins. Incidentally, such utilization also reduces environmental pollution. So there are microbes which are being grown on an industrial scale as a source of good protein. So the blue-green algae BGA, spirulina, it can be grown easily on material like wastewater. So from it can be grown on potato processing plants which contain starch, can be grown on straw, molasses, animal manure, even sewage. It can even grow, that is the blue-green algae spirulina can even grow on sewage to produce food which is rich in protein, minerals, fats, carbohydrate, vitamins, and incidentally such utilization also reduces environmental pollution. Certain bacterial species like Methylophilus methylotropus, because of its high rate of biomass production and growth, can be expected to produce 25 tons of protein. So other certain bacterial species like Methylophilus methylotropus, because of its high rate of biomass production and growth, it can be expected to produce 25 tons of protein. The fact that edible mushrooms are eaten by many people and large-scale mushroom culture is a growing industry makes it believable that microscopic fungi too would become acceptable as food. Okay. So single cell protein, that is, it is alternative protein that is alternative source for protein for human and animal nutrition. Microbes such as blue green algae, spirulina can be grown on wastewater and straw, molasses, potato processing plants, even on sewage to produce large quantities of and can serve as food rich in protein, mineral, fats, carbohydrate, etc. It will reduce environmental pollution. Even bacterial species such as Methylophilus methylotropus, because of its high rate of biomass production and growth, it can produce 25 tons of protein. And even edible mushrooms are such a growing industry that it makes us believable that microscopic fungi will also be acceptable as food in the future. Okay, now as traditional breeding techniques have failed to keep pace with the demand and provide sufficiently fast and efficient system for crop improvement, another technology called tissue culture got developed. So traditional breeding techniques failed to keep pace with the demand because the demand is very high because of the increasing population of humans as well as animals, and provide sufficiently fast and efficient system for crop improvement. That is, fast and efficient system to itna jaldi nahi milta hai, right? Conventional breeding techniques, even after like introduction of so many small sectors and all. Another technology, tissue culture, got developed. What does tissue culture mean? Tissue culture was learned by scientists during the 1950s that whole plants can be regenerated from X plants. That is, in tissue culture का base क्या है? Tissue culture में we get to know that a whole plant can be generated from an X plant. That is, what is explant? Explant is any part of a plant taken out and grown in a test tube under sterile condition in special nutrient media. This capacity to generate a whole plant from any cell or explant is called as totipotency. You know totipotent cells, right? Totipotent cells, that is in your placenta, that is embryo में जो हमारे totipotent cells होते हैं जिससे कुछ भी बन सकता है. That is, you can make any organ, you can make anything from those totipotent cells. That is, these totipotent that is under sterile condition, any part of the plant, that is an explant, that is any part of the plant taken in a test tube under sterile condition in a special nutrient media, it can regenerate a whole plant. You will learn how to accomplish this in higher classes. It is important to stress here that the nutrient medium must provide a carbon source such as sucrose and also inorganic salts such as vitamins, amino acids, growth regulators like auxin, cytokinin. By application of these methods, it is possible to achieve propagation of a large number of plants in very short duration. तो बेसिकली विद द हेल्प ऑफ टिश्यू कल्चर, दैट इज हम लोग एक्सप्लांट लेते हैं तो उससे दैट इज एक सेल से या एक पार्ट ऑफ द प्लांट से हम लोग एक पूरा प्लांट रीजनरेट कर सकते हैं, एन एंटायर प्लांट. एंड दिस एंटायर फिनोमिना कैपेसिटी टू generate a whole plant or a whole organism or whole human being from just a cell, that is totipotent cell, is called as totipotency. And for this the nutrient medium must have a carbon source such as sucrose, inorganic salt, vitamin, amino acid and growth regulators should also be present such as, that is, hormones, plant hormones, auxin, cytokinin, gibberellin, abscisic acid, ethylene, etc. And by application of these methods, it is possible to achieve propagation of a large number of plants in very short duration. You can achieve a large number of plants, and this method of producing thousands of plants through tissue culture is called as micropropagation. What is the method to produce all these thousands of plants by tissue culture? It is called as micropropagation, and each of these plants will be genetically identical to the original plant from which they will be grown. Therefore, they are called as soma clones. That is, these plants that will be produced from tissue culture, that is from tissue culture when we produce the plants, then all these plants are going to be genetically identical, and since they are going to be genetically identical, they are called as soma clones. Many important food plants like tomato, banana, apple, etc. have been produced on a commercial scale using this method, soma clonal method. Many plants such as tomato, banana and apple have been produced on a commercial scale. Try to visit a tissue culture laboratory with your teacher to better understand and appreciate the process. Now another important application of tissue culture is the recovery of healthy plants from diseased plants. Even if the plant is infected with the virus, the meristem, that is the apical as well as axillary meristem, is going to be free of the virus. Hence, one can remove the meristem and grow it in vitro to obtain a virus-free plant. This is also a very important and very amazing fact. That is, by tissue culture, that is from explant. Firstly, by tissue culture from an explant you can regenerate or generate an entire whole plant. The second part is that from diseased plants, that is plants that are going to die, we can recover healthy plants from diseased plants. That is, the meristem, that is apical as well as axillary meristem, is going to be free of the virus. So therefore, one can remove the meristem and grow it in vitro to obtain virus-free plants. Scientists have succeeded in culturing meristems of banana, sugarcane and potato. This is also very big breakthrough, you can say. Scientists have even isolated single cells from plants, and after digesting their cell walls, they have been able to isolate naked protoplasts surrounded by plasma membrane. Isolated protoplasts from two different varieties of plants, each having a desirable character, can be fused to get hybrid protoplasts, which can be further grown to form a new plant. These hybrids are called as somatic hybrid. Soma clonal plants are those plants. Soma clones are those plants which are made from tissue culture, that is X plant generated into a whole plant, genetically identical, that is soma clones. Now, so scientists can isolate the single cells of the plants, and they can digest their cell wall, and they can isolate the naked protoplasts which are surrounded by the plasma membrane. Now these isolated protoplasts of two different varieties, each having desirable character, can be fused to get hybrid protoplast. And when we grow this hybrid protoplast further, that is in the cell, that is basically an explant, right? So we can get a new plant, and this new plant is going to have the desirable character of both. So these hybrids are called as somatic hybrids, while the process is called as somatic hybridization. Imagine a situation when a protoplast of tomato is fused with that of potato, and then they are grown to form new hybrid plants combining tomato and potato characteristics. This has been achieved, resulting in the formation of pomato. Unfortunately, this plant did not have all the desirable characteristics for the commercial utilization. This is what I was saying, tomato and potato. That is, they thought that they would get potato inside and tomato ऊपर, but instead उनको उल्टा मिल गया. नीचे का वो tomato का part नीचे आ गया, potato का part ऊपर आ गया. तो इसलिए desirable characteristics नहीं मिले. So that is the end of the chapter. Animal husbandry, role in human welfare, no. Then if your family owned a dairy farm, what would you take? That is hygiene, good quality breed, all those things. ठीक है, अच्छे से इंस्पेक्ट करना, फिर इनको डिजीज रेजिस्टेंट रखना, डिजीज फ्री रखना, अच्छे से हाउस करना, वेटनरी विजिट ये करना. व्हाट इज द ब्रीड का मतलब क्या होता है? दैट इज रिलेटेड इंडिविजुअल्स ऑफ अ स्पीशीज, दैट इज एक ही 4 to 6 जनरेशन का सेम होगा, एंड वो एक ही इसके होंगे. What was that word? That is, they would be of related by descent. Yes, descent. They would be related by descent, and उनके बहुत सारे features, that is morphological and physiological, all features would be same. That is feature, size, general appearance, etc. Apiculture. Yeah, all these you can answer now. Okay, so it became a very big lecture. If you want, then I can discuss all the questions in the next year. Please do tell me. Thank you.